Hello, my name is Professor Matthew Schmidt, and welcome to Genetics. In this session, we're going to be discussing recombinant DNA, as we have been, but we're going to be focusing on its applications. That is, the real biotechnological things that we can do with this type of technology. Now, there's a certain amount of overlap because even within the previous sessions, if you've been watching in order, we said, okay, you can use restriction enzymes to clone a gene. That's an application. But now we're looking a little bit more broadly and saying, great, you can clone a gene. What do you do with that? Or, what are the more, I guess, everyday, I don't know if it's everyday, but everyday use, practical type of things that we can extract using these basic technologies. So let's take a look. And obviously, this is not going to be an exhaustive list. We're going to focus on the ones that are the most common and the most used. So here we just have a list, applications of recombinant DNA technology. Firstly, identifying and isolating human. Now I wrote disease genes here, but think about this. If you've been following along, one of the big examples we used was identifying and isolating the human insulin gene, right? And then we already showed how, basically we showed how you can use that to actually make human insulin, which technically should be in this section, but we were using it as an example. So it doesn't have to be a disease gene. It's just that obviously genetic diseases, we certainly want to know what gene is responsible. We want to be able to know what it does and to characterize it, etc. So it's not limited to that, but certainly disease genes are widely studied. And a lot of times, as we're going to see, we learn about the normal function of genes by looking at the disease gene. We say this person has this problem. Oh, look, this gene is altered. What must the normal, if you will, version of that gene do? Once we have that information, it's a little controversial to some people, but we can test for genetic diseases. I wrote prenatally here. Sometimes that's desirable, sometimes later in life it's desirable. A big one that I know you've heard about has to do with so-called DNA fingerprinting, which can be used for a variety of things, but the most common are paternity testing and forensic analysis of biological material. Commercial products, as we just mentioned, so making recombinant protein substances using bacteria. Human insulin is a great example. Human growth hormone is a protein, so you can make that as well. So a lot of things that are, I should say, some things that are traditionally, I mean in the past, insulin from different animals was used. Now we can make it easily. HGH could be extracted from humans, but now we can make it in unlimited quantities. Gene therapy is something that we really hope is going to be a major thing. I'm not trying to minimize it, but the actual successes with it have been a little bit spotty. But this is the idea of saying, wow, you have a genetic problem, maybe we can do something to fix it, or variants of that. Genetic engineering, I know you've heard about. That's another sort of broad term. I mean, in one sense, engineering means any manipulation of genes, but in this sense, I'm talking about transgenics, which means taking a gene of interest. We've done something like this before, from one species and putting it into another species, or changing a gene that's in another species. So those are very interesting things. And cloning of organisms, we've talked about cloning of genes, but of course you've all heard most of it's science fiction about taking oneself and making a clone of it for various purposes. I mean, if I could do it, obviously the world would be a much better place with a bunch of me's running around. Just kidding. But it is, a lot of these things have ethical components to it, which we can't cover in great detail. Bioethicists have to do that. Maybe that's a field you might be interested in. But certainly it would be controversial if we had the capability to clone a human being, depending what the idea was we were going to use that individual for. So let's take a little bit more detailed look at each of these. So the identification and isolation of human genes focus on disease genes. Now we already know that recombinant DNA techniques in general will allow us to map a particular area of interest on the DNA. But mapping means a couple of different things. So think of a human, let's say it was a human disease. You would want to know a variety of things about the gene. If you could identify the gene, where, what chromosome is it on, for example. And then you'd want a more fine scale map, maybe like a restriction map showing where this gene lies in relation to the other either genes or other parts of the chromosome, because as we'll talk about in a different lecture, most of the genetic material is not necessarily genes. And ultimately, if you're able to pinpoint exactly where it was using techniques we've discussed, you can do a DNA sequence. Now, you could sequence the mutant, in other words, someone who is suffering with the genetic disease, compare it with the wild type allele, meaning that which is derived from someone who's healthy, who doesn't have the disease, and you can see what's the difference between them. It wasn't done this way, but we've discussed a while back how hemoglobin from someone who had sickle cell anemia is not all that different from normal hemoglobin, but we know exactly where it is different, and that explains the difference in the protein and the disease. So a lot of times it's really not, sometimes it is, but it's not always that obvious just from looking at the physical manifestations or the symptoms of a genetic disease exactly what type of gene is going on or going wrong. Cystic fibrosis is a perfect example of that, where there's a problem with epithelial function, mucus overproduction, etc. And it turns out that it's an ion transporter that's the gene that's being compromised. So interestingly, if you can do this, now I said this allows prediction of the sequence of the wild type protein. So in other words, you have to maybe think about this for a minute, but using techniques we've discussed, you can isolate a gene and not even have any idea, assuming that it makes a protein, which it probably does, what that protein is or what it does. Once you know the gene and the wild type sequence of it, you can predict just from the sequence of the codons the amino acid sequence of the wild type protein, right? And then you can, there's a database that we're building up. You could say, okay, well, this protein sort of resembles, just for example, another one we know is a transmembrane protein. Then you can narrow it down. So it can give a clue to what type of a protein this is, what it probably does, and then you can try and get to the bottom of what the normal function and the aberrant, the disease form function is. In other words, what's changing in this protein, just like we mentioned about hemoglobin, that's causing the disease. But remember, the reason the hemoglobin is different in someone with sickle cell anemia is because they have a mutation at the gene level, at the DNA level, right? So I'm saying this is a prerequisite to gene therapy in the sense that we'll talk about gene therapy in a minute, but the bottom line is if you want to replace or alter the functioning of a gene, clearly you have to know what that gene is and have some idea of what it's supposed to do or what it's doing wrong or what it's not doing at all. Okay? Now, we've been successful in characterizing a lot of genetic diseases are caused by a single gene mutation, a single mutant. Not all are like this. Just as an example, Down syndrome is a chromosomal excess. There's an extra entire chromosome, right? So that's not one gene that's causing it. In the things that we're talking about now, it tends to be one gene that's either solely responsible for the disease or predominantly responsible. So here are two sort of popular ones. Huntington's disease. Now scientists who discover the gene pretty much have latitude in naming it whatever they want. They've called this gene Huntingtin, which is spelled a little different. I honestly don't know why. Now, this is not something you'd have to memorize, but it's something you would want to know if you were actually working on this. So they've mapped it to the short arm of chromosome number 4. And then getting much more specific, we have sequenced it and found out that we know the wild type sequence. And what's different in people with Huntington's disease are these trinucleotide repeats in a high copy number. So whatever it is, I don't know what it is, say it's ATG, you have loads and loads and loads and loads of extra ATG repeats, sometimes the more severe the disease is, but we can't get into that. So, and check this out. We can tell by the genetic analysis. There's a test for Huntington's disease. This is a disease that's interesting because it doesn't manifest usually until later in life. It could be anywhere between 25 and 65 that you start having symptoms from this. You've had the gene from birth though, right? So, you know, one of the interesting things about this is if one of your parents starts exhibiting the symptoms, or if you know there's a family history, there's a much greater chance that you in fact are going to have the allele. You can do a genetic test and you can know just like that. Now this is one of the cases where even though we know the sequence of the wild type protein, I should say the gene, and therefore can predict the wild type protein, we really don't know exactly what the Huntington protein does. I mean, Huntington's disease, it's related in a certain sense to like Alzheimer's and other dementia type diseases. It results in a degeneration of the nervous system. So we don't fully understand that. Perhaps obviously the brain is probably the organ we understand the least, and the genes that work to make a properly functioning brain, that's something that's very, very complex. However, we can still tell if you have it or not. And you can't say with 100% certainty, but if you have the allele, there's a pretty good chance, the mutant allele, that you will develop some aspect of the disease. I mentioned cystic fibrosis before. They just named the gene CF for cystic fibrosis. Again, you wouldn't have to memorize it, but this one happens to be on the long arm of chromosome 7. This is just another illustration. There's really no particular rhyme or reason in eukaryotic genomes, the human included, of why a particular gene is here or there on this chromosome or another. Now, when they started looking at people with cystic fibrosis, they found all sorts of mutations of this one particular gene, though. Over 100 have been identified, and again, most of these we now know are lack of function mutations. In other words, changes that don't allow the protein to either function at all or probably allow it to function rather poorly compared to what it's supposed to do. So we have found out that the normal wild type protein, it's named because it was discovered, as I mentioned or implied before, it was discovered only by analysis of the disease mutant, I should say the mutant that causes the disease, and then we found out the regular gene that everybody has. So it's called the cystic fibrosis transmembrane conductance regulator, a very lengthy name, so most people just call it CFTR, but it's an ion channel which controls the transport of chloride ions in epithelial cells, particularly respiratory epithelium and digestive tract epithelium. So we all have this gene, in other words. It's a regular ion channel. If it's not able to function properly, I mean, who would guess if I just said, gee, this chloride transporter isn't functioning properly? I doubt anyone would guess all of the symptoms of cystic fibrosis just because of that. So we have to work backwards, and in this case, we know exactly what it is. So anything that's preventing that protein from functioning properly is causing symptoms of the disease, which can be very severe or less severe. All right, so with this type of knowledge, and you could go on and on, there are several of them that have been identified, you can say, I know what the wild type's supposed to look like. I know what either the mutant or many popular mutants look like. You can design tests also using the technology that we've been discussing. For example, PCR based on length. If it was Huntington's, right, the mutant allele would be a lot longer than the wild type allele. But regardless of the different methodologies, there are many tests that have been designed to diagnose genetic diseases. So Huntington's and cystic fibrosis we mentioned. Fragile X syndrome is a problem with the X chromosome that causes mental retardation to varying different levels. It involves also repeated units that cause the chromosome to become different size. We can tell that, and many more. Now, very interestingly, and this is where some of an ethical thing comes in for some people, A lot of these tests can be performed prenatally. Now, just take in the case of cystic fibrosis, the parents could be tested, and if you'd have to have both of them being carriers in order for the child to possibly, in fact, get the disease. But you know from way, way back when, it's uncommon, but with all the millions, billions of people around having children, very, it's not terribly uncommon that two people will happen to meet and they'll both be carriers of the cystic fibrosis mutation. In that case, you guys probably know, well, since it's a recessive disease, the probability of their offspring having it is 1/4. And potential parents are faced with a big problem and a big decision here. You have 75% chance of having a normal child and 25% chance of having one who's going to have the disease, whose life may not be as pleasant as it might be. Why is this an ethical issue? Well, some people wouldn't and some people would, but sometimes if you know there's a genetic problem with an embryo or a fetus, it's possible and it's legal in many cases to abort that pregnancy. I'm not making a value judgment about it. I'm just telling you that's why some individuals don't even want to do the test and why there's some people that this makes feel uncomfortable. On the other hand, some people just want to know, and I guess you can draw your own conclusions. Very, very interestingly, and this is ethically interesting in a different way, a lot of times, I probably mentioned this probably a few times, my daughter was an in vitro fertilization. What they do is they usually have multiple embryos around and they decide which ones they're going to implant. It's not routinely done, but if there's a good reason to believe that there's a genetic problem, what you can do is, it's not easy, but it can be done. You can test those little embryos because they don't need, I won't go into all the details, but if you find that two of them had the cystic fibrosis mutation, you wouldn't implant them. You would implant ones that are healthy, as it were. That's a little bit different, right? That's saying, look, you could have had some offspring that are affected and some that aren't affected. We're pre-screening them and we're just going to implant the healthy ones. That's less objectionable to some people, but some people just don't like the whole idea of it. But it's, aside from the ethics, it's obviously fascinating and wonderful that we can do these things. So genetic counselors sometimes just use family histories and pedigrees, but if these tests are available, they can advise individuals about their risks or those of their potential offspring, or even actual, if they're having these type of molecular test results available. All right, so that's one very important part of it. Let's move on to DNA fingerprinting. Now, DNA fingerprinting is a technique, but what it really is a modification, or I guess I should say an application of the Southern blotting procedure that we went over. So the whole idea of DNA fingerprinting is based on the fact that there are polymorphisms in human beings. Polymorphism is just a fancy word for an alternate shape or alternate form. It just means there's different alleles at different parts of the human genome. I mean, we know this already. Maybe at the eye color locus you got blue or brown. But they specifically select places in the genome where they know there's a lot of variation. In other words, certain non-coding regions, there may be 100 known differences. I might have one of them, you might have a different one. And so the bottom line is often these are length polymorphisms of repeated sequences. So if I have one repeat and we do a restriction enzyme cut, I'm going to have a fragment of a certain size. Someone might have 40 repeats, someone might have 60. So the bottom line is, I say here, it translates into some bands on a southern blot, because remember you're probing with something of the gene of interest, which is going to bind to whatever's on that southern blot. They're going to be lighting up, as it were, bands of different sizes. And you can run that out in you already ran it out in a gel and you can tell. So officially, DNA fingerprinting is thus called restriction fragment length polymorphism analysis, RFLP. Just think of what you're doing. Remember what a southern blot is. First thing you do is digest everything with restriction enzymes, right, to make a bunch of fragments. And if their lengths are different from person to person, that's what you're getting at. So let's say you wanted to do either a paternity case, or even though hopefully it's rare, everybody's interested in crime scene, DNA analysis to try and I'm not. It's very important. I'm just saying. I hope it's not an everyday activity for most of us, unless that's the field you're in. Another good one to join. So forensic analysis, paternity cases. Let's see how that would actually work. See, we've already done the thing about the southern blot. I'm showing it to you here again, but we want to focus on one portion of it. So just remember what you did. Let's say this was a paternity test, okay? Let's say these 3 samples were actually 3 different individuals that were putative fathers. We would take samples from each of them, run them down 3 different lanes of a gel, right, in an electrophoretic field, blot, apply our radioactive probe, and then remember we wash it off, and we end up with a gel that looks like this. Now this, I'm going to show you a different picture in a minute, but the point here is that in some lanes, like individuals 1 and 2, seem to have bands in the same location, whereas individual 3 has more bands in different locations, so you can differentiate them that way. But the point here is not, we've already gone over how to do a southern blot. The point here is how you actually apply it to this type of analysis. So again, just say it's a paternity test. There's a sequences, and I was just referring to them. They're called VNTRs, variable number tandem repeats. So I was just saying, these are spots in the genome which, for whatever reason, I could have 10 repeats, you could have 30, someone else could have 90. When we cut with certain enzymes, they're going to reveal different length fragments for different people. So the name implies different people have different copy numbers, and that creates restriction fragments of different sizes. So in paternity testing, this is sort of interesting. We have the mother and the child, and I'm not being sarcastic. The mother pretty much knows that the child came out of her. There's not a lot of doubt usually who the mother of a child is, right? Unless really weird things happen. But we can get DNA from the mother and from the child and say there are 2 potential fathers that we want to check out which one really is the father. Now, think of this. The child has to get half their genes from the mother and half from the father. And since we have the DNA from the mother and the child, there's a question mark about, all right, we're going to be able to see probably what it got for the mother, which father matches the other thing that it has. So if you do a southern blot, now I just drew this, it's not as fancy as some of the other drawings, but here's going to be an autoradiograph showing the results, okay? We have an electrophoretic, it's not a gel anymore, right? This is the blot, but originally we loaded all the samples here and they were traveling in that direction. And the smallest ones would go the furthest, and the largest ones would be not traveling quite so much. So here's what we got to do, guys. We look at the child and we say at this particular locus, whatever it may be, this child got bands of, and this is the simplest case, but it's not that difficult to do this. The child has bands of 2 different sizes, right? It had to get one from the mother and one from the father, right? Well, perhaps clearly it got that one from the mother, right? Because it didn't get that one from the mother. So the question is, where did it get this one from? And the answer is very clear here, although with a little qualification. Most people would say, well, look, the only one of F1 and F2 that could have donated it was this guy. So this guy's the father. But here's where it can get a little bit confusing. What this really does is it rules out candidate number 2. It does not prove that F1, potential father number 1, has to be the father, because think of this. Out of those 2, he's the most likely candidate. But how do we know how many other individuals in this world or in a particular geographical or ethnic population have this same pattern? The guy could say, assuming he didn't want to be the father, or it could work either way. He could say, look, I have the thing, it's me. Meanwhile, 10% of the population has a similar situation. So how do we do this properly? I shouldn't probably mention the name, but there's a certain TV host about, seems like every time I see the show, it's about who's the father, and they always come out with definitive proof. You are the father, you are not the father. This is based on the idea that two ideas. One of them is that there's a pretty small, usually it's either this guy or that guy. But here's the other idea that goes behind it. And with forensic analysis, it's obviously a lot more complicated because you don't necessarily have it's either this guy or this guy. It could be all sorts of people. So how we really do this? Let's say the allele that we're looking for in this diagram here, I mean this one. This is the one we need to know. Somehow the child got that from some father, right? We have one potential guy here who could have done it. But maybe that allele is shared, as I just said, that's shared by the child and the father, could be present in like 10% of the population. So if you lived in New York City, that could mean 1 million people could be potentially the father of that child, right? That's not that great. So the data does rule out the person that we called F2, in other words, potential father suspect number 2. But what it says is that there's only a 10% chance that the shared allele is simply a coincidence. But 10% is still a pretty big margin of error. So I can't go into a lot of the details of this, but they use a multiple probe system. 5 or 6 different places in the genome where there are these variable number terminal repeats. So each time, so they'll do that. They'll say, okay, let's check this guy out more. Let's use a different probe. See if he matches the child at that area. Let's just say it's an oversimplification, but let's say it's always 10%, right, of the population that has a particular one. But each time they match, the odds of the coincidences are lessened. In other words, if you have at 2 totally different places in the genome, if the father shares those 2 with the child, now it's not a 10% chance it's coincidence, it's a 1% chance that it's a coincidence. And if you use 5 probes, you can get the odds down to 1 in 100,000. In real life, the way that they do it, you can get the odds down to 1 in 10 billion. Now, there's only 7 billion people on the world. So if you say, look, there's a chance this is a coincidence, but it's 1 in 10 billion, as a friend of mine who worked in the coroner's office once said, in other words, it's him. But you have to be careful, and lawyers, no offense to lawyers, they'll argue this. It does not prove it. What if the guy, and it may sound silly, but this is the basis of a lot of books and soap operas, what if the guy had an identical twin somewhere? It may be a 1 in 10 billion chance, but his identical brother probably is the father. You never can prove it 100%. That's why, guys, I'm not a attorney, perhaps obviously, but they never say you got to prove it. They say you have to be convinced beyond a reasonable doubt. And the system isn't perfect, but if there's a 1 in 10 billion chance that this is coincidence, usually that's enough to convince almost everybody that this is the person, especially if there's other evidence, forensic or otherwise. But hopefully you get the idea. It's a simple idea. It comes down to a lot of statistical analysis in the end. Okay, we're moving right along to gene therapy, something we wish we could do a lot better than we do. The idea is pretty straightforward. That idea being to treat genetic disease in a way that we never could before by literally replacing a faulty gene with a normal one. Now, this is not the only way, but this is the most straightforward and easiest to understand. Say you had cystic fibrosis. We know that there's a problem with that CFTR protein, and that means there's a problem with the gene. What if we can either replace it or supplement it with proper copies of the gene? So we take some wild type alleles to either replace or to supplement. Because supplement, in other words, say you're a hemophiliac. You're not making enough clotting factor. You don't necessarily have to get rid of the faulty genes. You do need to incorporate good ones that will be able to make a clotting factor. In the case of cystic fibrosis, it would be more desirable to try and get rid of some of the old ones, or all of them, and replace them with new ones. Now, if you transfer a gene like this, it's called a transgene, and if that was a successfully done treatment, you would become a transgenic individual. Now, usually that's a transgenic mouse, for example, has human genes often put into the mouse, so that's a cross-species transgenic. But this means you're getting a disease that is not you're getting a gene that's not naturally part of your genome, so it's technically a transgene. Now, it's easier said than done. Another approach uses either antisense RNAs or ribozymes, which I think we've discussed a little, but an antisense RNA often binds to a messenger RNA, for example, and prevents its expression. That's often done when we want to stop a gene's expression, not augment it. So, in other words, say in AIDS or cancer, there are genes being expressed we'd like to shut down, and using antisense RNAs or ribozymes, which are enzymatic RNAs, they chop up RNAs so that they won't be expressed. Obviously, this is a big field, and you could go into a great amount of detail about it. But I guess the real quick version would be you'd like to do this sort of thing if you want to express a gene that's not being expressed or not being expressed properly. You generally want to do something like this. And when I say use antisense RNAs, you could still put the gene for the RNA in there. It doesn't mean you're actually using RNAs, although you might. You do this if you wanted to inhibit the expression of a gene when the problem is its overexpression. Now, that's a pretty simple idea, but how do you actually do it is the thing. So there are a lot of possible ways that people have thought of to maybe get a gene of interest. Think of it, this is not so easy as popping a gene into bacterium. If I just inject some DNA in my arm into my bloodstream, it's not going to get incorporated into any place that it needs to be. So ironically, many people think retroviruses, which can make you sick, have emerged as a prime model and a mode of gene transfer. The idea being that if you I'm sorry, I don't know if we've discussed this and it would be more a microbiology topic, but retroviruses, their whole thing is that they're RNA viruses. They use reverse transcriptase to make a copy of themselves as DNA, and they insert themselves into your genome, and then they usually express the bad disease-producing genes that they have. But we can engineer the retrovirus, take out the things that make it virulent or pathogenic, and put in something that we want. It doesn't know. It's a little robot, a very good one. But in other words, the idea is to say, okay, retrovirus, go infect the specific cells that you infect, but you're going to now deliver something that we want you to deliver, and you're going to automatically insert it right into this person's genome. Now, there are some technical issues, many technical issues with this. Some success has been reported, but it's limited, but we really think that the promise for the future is great. Some diseases where there's been some success is severe combined immunodeficiency. This is almost like an inherited version of AIDS, but it's even worse just because your immune system barely functions from birth. Cystic fibrosis, they have had some success. It's not all the details, but they actually shoot the genes into your respiratory system in the hopes that, in large quantities, in the hopes that they'll be taken up. I don't mean it's like gee, I hope. They do everything that we can do to try and allow that to happen. It's almost like don't take this literally, but it's almost like a transformation, right? Where bacteria pick up DNA that's out there. We're hoping our cells are going to pick this up. If you really were to do this, there's loads of technical details. Alzheimer's even, hemophilia, we've had limited success, but the day the success is ridiculous and just cures something like that, you're going to see it on the front page of newspapers if they still have newspapers at that time. So here's a pretty good illustration of one way that you could do it. The way I was just describing it, like using a retrovirus, would have to be considered in vivo, right? Because we're literally putting the new gene inside of a cell in the body. Ex vivo gene therapy is thought by many to be more practical. It really depends on what you're trying to achieve. But look what happens. So here's a child. What we're going to do is remove some stem cells from that child, right? Because stem cells are easy to propagate and they should be immortal. These stem cells have a defective gene in them. Now, look what we're doing. We're doing pretty much what I said, but we're doing it outside the body. This might reduce some of the potential side effects. So we're infecting these stem cells with an engineered retrovirus, which has the normal gene. Reverse transcription makes that viral RNA into DNA, and we're hoping that the normal gene is going to integrate into the genome, either replacing or augmenting the faulty one. Then we take these these cells that we took out of this patient, and we put them right back into this patient. This minimizes the possibility that there'll be an immune rejection or something of that nature. It's just that it's easier to manipulate these particular cells and know that they've been changed than just shooting retrovirus into the body. So this is a more targeted approach. But it really depends on the nature of the gene, the disease, etc. But if you get this idea, it's pretty powerful. And I know I mentioned the term transgene and transgenics before. In the more traditional sense of that term, transgenics refers to introducing foreign genes, meaning usually those from a different species, into plants or animals. It's funny, when you take the human insulin gene and pop it into bacteria, they usually don't say that that's now a transgenic bacteria. They say it's been transformed. When you're using multicellular organisms, the convention is to refer to this as a transgene. So transgenic organisms, plants or animals can have them, can be them. It can be done for a variety of reasons. So I'll give you a couple of examples. If you want to study a gene's function in vivo in model organisms, including disease genes, it doesn't always work perfectly, but let's say mice. Mice are a great mammalian model. There are all sorts of mice running around, hopefully not in the wild, but you never know. This makes some people nervous, but a lot of times we'll take human genes and put them into the mouse. So in other words, here's the mouse. The only thing different about it and its brother or sister mouse is that it's got this human gene in it. And then we ask, so what happens? What's different about this mouse? Either a physical trait, or we can biochemically assay. Does it make more of this enzyme, or does it do this? This is a way of figuring out at a fundamental level what the heck this gene does. Or we could put a normal one in one mouse and a mutated one in another mouse and see what the difference is. So transgenic mice are very popular. Organisms that produce novel products or have altered genetic properties can be generated. So for example, plants can be manipulated, and again there's some controversy about this, by sticking a gene in them. It's either usually an engineered gene. One common one is that, and this is very controversial, plants that are so-called Roundup resistant. Roundup, you may know, is a chemical that generally is a weed killer and it kills all plants. But what they've done is genetically engineer, by this method, plants that are no longer affected by Roundup. In other words, they're Roundup resistant, almost like some bacteria are antibiotic resistant. So this allows you to just spray Roundup all over the place. It kills, the idea being it kills weeds and other junk while not affecting the plant that you want to grow. But without going into all the details, some people are nervous about either eating, these plants or the spread of the transgene through the ecosystem, etc. Again, it's important to recognize, but we can't be bioethicists. We can be, but we more want to understand what's going on here. So here's an interesting picture just to show you the results. We have a transgenic mouse on the left that has actually not been injected with human growth hormone. It's been changed so that it now actually has human growth hormone gene in its body. And as you can very clearly see, that's the only difference between these two mouses. This one over here has, I don't know what that shape is, has the human growth hormone gene in it. Understand, I know, you can inject human growth hormone into your body and it can have effects, but I'm telling you that's not what happened to this mouse. It actually was engineered as an embryo to have the human growth hormone gene in it, and as it grew a lot larger than its friend over here. So that's just one example of a transgene and a transgenic mouse. Well, we could go on for a really, really long time. You could have a whole course in this subject. So my goal here really was to show the basic tools, the basic methodologies, and then to show ways in which they can be applied in a, I guess what you would say, applied in a practical way to a lot of different situations that are technological, from disease issues to forensic issues, etc. Well, any of these topics that you're more interested in, I'm sure you can do some more research about, and I will see you real soon at another genetics session.